Perfect. Thank you so much. Just a quick sound check and you can see the slide, main slide.
Yes, we can see them.
Perfect. Excellent. So thank you very much, Chiara, for the very kind introduction. And I'd like to start actually extending my thanks to you and the organizing committee for giving me the opportunity today to talk to you today about drug design and properties. It's also great to see Alessio again and be on the side of Luc for a great series of talks. And I'm looking forward also for the panel discussion. So here's my disclaimer. I prepared this lecture a little bit with a heterogeneous audience in mind. So you will hear perhaps some of the basic property groundwork as well as some of the advanced. So I hope there's something in for everybody. But please, if it's too basic for you, wait for the second part of the talk. I think I'll extend a little bit further. So I'll start by discussing what all this fuss is about physicochemical properties. And we'll be moving on to lipophilicity as the core concept of how we can put design into our molecules. And I'll share with you a case study on PI3 kinase delta inhibitors, and incidentally a vignette on gamma T inhibitors where Luc also had his little story. And in the last part, I will be moving on to forecasting properties. How can we predict what we make? We'll start with the famous rule of 5. And as this is quite around for quite a while, have a look of where we are today. And then in the context of today's main topic, what happens if we go beyond? What happens if we go to molecules with higher molecular weight? So medicinal chemistry is an art form of arriving at a clinical candidate that it's in its most essential form is only needs to fulfill 2 requirements. It needs to be efficacious and it needs to be safe. And of course, there we need to work on various different areas. So on the efficacious side, we need to make sure that the compound can exert its therapeutic effect. It needs to be potent and functional on the target. We typically assess this in biochemical cellular assays, cell assays, functional cell assays, cellular assays. And then we need to efficiently get to the site of action. So the pharmacokinetics of the compound plays a major role, and I will get to this in much more detail. So high potency of a compound plus good pharmacokinetics typically leads to a low dose, and low dose is arguably the one main predictor of whether a compound will make it through clinical development phases successfully. But of course, a compound also needs to be safe, so needs to have no effect on other targets, so other isoforms of the protein of choice, for example, within the target class, and other safety relevant targets, again assessed by various biochemical and cellular assay systems. And again, a low dose helps typically to get on the safe side. So as I mentioned, the art form is actually a multi-parameter optimization. This is also what Alessio mentioned and Luc mentioned to arrive at a good candidate. And today I will focus mainly how to efficiently get to the site of action, how to dial in the properties that we need in successful drugs. But first of all, let me enter the area by saying there's, of course, different routes of administration. And whilst the intramuscular route is now in everybody's minds as we talk about the pandemic and vaccines, it is still the oral route that is probably the main driver of drug discovery, as people still prefer to swallow a pill rather than have a needle injected in them. We differentiate between the enteral and the parenteral routes, as you can see here, and these are the main ones. But of course, there are others. And for today's purpose, I will focus mainly on the oral route for the reasons I just gave. And if you think about it, the challenges to a molecule needs to phase once it gets swallowed as a pill to its intended site of action are numerous. So first of all, it needs to be digested in your GI tract. Then it needs to get absorbed across the gut wall cell membrane. It will be taken up by the portal vein transported into the liver, where it's first is metabolized for the first time, and only then it is released into the bloodstream. Obviously, then makes it to the site of action, passes multiple rounds via the liver, which is your main metabolism detoxification organ, and is ultimately excreted. So when we talk about molecular properties, we often refer to them as ADME properties. Many of you will have heard that. That's short for absorption, distribution, metabolism, and excretion. And I'll start with absorption. So what are the requirements for good absorption? We are here talking about digesting a tablet or a compound. So first of all, it needs to be sufficiently stable in the GI tract, and then it needs to show a sufficient dissolution in the GI tract. And dissolution is modulated by two parameters. First of all, it needs to have a reasonable rate to be dissolved, and then it needs to be soluble, which is basically a capacity issue. And again, the GI tract has different pH values and different lengths. It needs to pass through the main areas, the jejunum and ileum, where the drug passes over 5 meters in length, slightly basic typically, and there the main absorption takes place. But of course, the compound also needs to move across membranes. So they need to have sufficient uptake through the gut wall. So membrane permeability plays a main role there. And the third limitation and barrier to absorption is the hepatic first pass metabolism, where the compound is being taken up by the liver in the first place. And of course, all of these absorption problems you can shortcut by an intravenous injection, and many compounds, as you know, are actually IV drugs. And I would argue, even though we often talk about ADME properties in industry, we can fundamentally only influence absorption and metabolism really well. We have certain means for distribution, that is true, but to excretion, I think we're largely bystanders in drug discovery. So what I will mainly talk about is solubility, membrane permeability, and to some extent metabolism. These are the main areas where we can really do something about in medicinal chemistry. So one of the main questions is, should we be hydrophilic with our drug molecules or lipophilic? And with respect to aqueous solubility, which we need, as gastric fluid is largely, of course, water-based, we need to be quite hydrophilic in order to have molecules that dissolve there. And we can assess this in various assays, kinetic and thermodynamic solubility assays. You will find some of the brief outlines of those assays in the brief handout that you see in the chat. With respect to gut wall permeability, the transcellular route that you see over there, where it needs to pass two membranes, so the outer membrane and the inner membrane is the main part, the predominant part. And of course, here the lipophilic molecules have a better chance, as the lipid bilayer is of course lipophilic and easier to pass for lipophilic molecules. And we've got a number of in vitro assay systems how to assess that, and here are a few listed, again, details in the handout. And then to get to metabolism, so if you think about it, the liver, the body's detoxification machinery has been actually has evolved to metabolize mainly the lipophilic molecules. So it's designed to make them more polar and to make them amenable to excretion by, of course, the aqueous bodily fluids. So again, the hydrophilic molecules are metabolically more stable, and the lipophilic ones are going to be more prone in general to metabolism. And we've got in vitro assay systems to assess that, typically turnover in rat liver or human liver microsomes or hepatocytes. So ultimately, the question to go hydrophilic or lipophilic is not really valid, because you need to, of course, have both in your compounds. And it is, in a way, in the analogy you see, to be like walking on a ridge. You see this mountaineer has to come over here in order to stay safe and not fall down one end or the other. You need to be in an optimal range and walk the, what I would call the lipophilicity range, in order to be successful there. If you are too hydrophilic, or if you make a molecule more hydrophilic, you will typically, as I said before, make it more soluble, make it more stable in terms of metabolism. Typically, you will notice that membrane permeability goes down. If you go in the hydrophilic way, you will encounter the opposite. But of course, there's other areas that you also need to take into consideration. If you are too hydrophilic, you will notice that your on-target potency also quite typically goes down, unless you have a targeted interaction. On the plus side, also your promiscuity, so your compound binding or affinity properties towards unintended proteins will go down, which works in your favor. And again, this is the opposite as you go in the more lipophilic arena. And this arena has often been termed molecular obesity, in analogy to being too lipophilic. And I will now outline of why this is. Why do we typically go into this area more than we go into this area here, where we should err on the side of caution. And the reason is that we typically want potent molecules on our protein targets. And if you think about what's necessary in order to bind sufficiently to a protein surfaces are typically more or less lipophilic, or many protein surfaces are lipophilic, I should say. And when you take a molecule and move it into the protein cavity, it first has to be desolvated. So you need to move these water molecules out in order to make it available for binding in the protein cavity. And there's an enthalpy of binding which works in your favor, but of course you have the dissolution penalty, which typically works against you. So this is why typically, if you are more hydrophilic or too hydrophilic, you will notice that your KD goes down, and this is to some extent down to the dissolution barrier that you need to overcome. And of course here, after binding, you also release water molecules from the protein cavity. Again, this is an oversimplification. There's also other entropic considerations that you need to play into account, which are largely not well understood with respect to their overall strength and impact on the overall process. So to get back to our obesity analogy, it is typically the easy way out if you don't move too much. If you sit on the couch after hard days of work rather than go for a round of exercise, it's also our sedentary lifestyle that quite often demands this. And again, coming back to pharmaceutical industry, it was really interesting to see in 2011 a study where the AstraZeneca scientists looked at the differences between compounds that were designed in the 2000 to 2005 arena as compared to the 2006 to 2010 arena. These are the two bars that you see. And in general, there are companies that largely kept lipophilicity around the compound series the same as measured by the logP, which I'll get to in a moment. But there's companies like Takeda here or AstraZeneca at the right that really made quite an effort to get to compounds with reduced logP, which is a lipophilicity measure, as I will outline. And many companies perhaps will not have made great progress. And other than scientific reasons, I would say there's also behavioral reasons there. There's certainly also a company factor in there, perhaps also mediated by metric-driven goals, where making more molecules is sometimes seen as more advantageous rather than the hard route, which typically is to make them less lipophilic. Well, I mentioned C-logP or logD already. This is perhaps the one parameter to rule them all, to stay with the Lord of the Rings analogy. Well, it's not quite true. It's nearly all of them, but certainly logP or logD is the key parameter that's involved in all property desirability scoring systems. And here you find a list of them. There are more, and I will touch on them a little bit later in the talk. So what is logP or logD? So logP is the partition coefficient, and logD is the distribution coefficient. And it's actually, if you partition a compound between a layer of N-octanol and water. So N-octanol, a lipophilic liquid that is supposed to also, to a certain extent, depict the membrane. You will find a certain distribution between these two liquids. In its neutral or unionized form, it will be the logP is defined as the concentration between the octanol and the water layer. In terms of neutral molecules, the logD equals the logP. For charged molecules, the logD is typically lower. You measure that logD at a certain pH range, and if you consider a pH value or a neutral pH value, which is where it's most relevant, the logD, the higher the pKa difference is from your pH value, so that the more acidic or the more basic in the basic sense it is, the larger this term here becomes. So you take more of the logP, so you drop the logD to a lower degree, the more the pK is different from the pH. So again, a high an acid with a very low pKa will have a very lower range of logD as compared to logP, which is typically where you want to be. There's different ways to determine the logD or logP. One of them you saw on the slide before, which is called the shake flask method. Basically, you measure the concentration in those two liquids. There's also the chromatography determined logD, which is something that was developed by scientists at GlaxoSmithKline. And here you see when they plotted their chrom logD versus the normally determined logD, they saw that it sort of tails off here. So the compounds that were determined with a logD of 3 were either over or under predicted, or equally right with the chrom logD. You could span that a little bit out and decrease the dynamic range here, largely contributed to lacking solubility in this area. They can also predict this quite nicely, as you can see on the right hand side. There's also I need to stress that different ways to calculate logP. So C logP is sometimes not C logP. There's different ways to calculate that, just to make you aware of this. But wherever you can, I would argue you should determine your logP or logD experimentally, as it can turn out in this real data set of PI3 kinase delta inhibitors, a story that I will get to in a moment. It could be that you see logP either under predicts, as you can see here, or you see logP over predicts, so that the black line is the line of unity. And the color code is by pampa permeability in a traffic light format. And as you can see, what you would expect is in the high hydrophilic area, you start to observe problems with gut wall permeability as predicted by the pampa assay. So what is the optimal logP or logT? And there's many publications which I outlined below. Again, all the publications can be found in the handout. No need to take screenshots. There is a so-called sweet spot between logP 2 and 3. So this is a long-term goal to stay in that area. However, I would argue that sometimes you can even get lower. So the way I like it much better is to actually go as low as you can without affecting your target potency and without hitting your membrane permeability limit. You find this, in other words, here. So this is probably where you need to strive in order to get your molecules into the right property space. There's also a concept around lipophilic efficiency or LipE, sometimes also abbreviated LLE. So this concept puts the activity you have on your target in the context of lipophilicity, and this is nothing else like the negative log of your IC50. So this is the PIC50, and you take off your logD. In other words, if you increase your lipophilicity of your compound by a modification by 1 log unit in logD, you would need to increase your potency by a factor of 10 to stay in the similar lipophilicity efficient area. And there's many critics of the LipE as a concept, but I quite like it, especially if you look at it in evaluating progress within one distinct chemotype or chemical series that you monitor into an optimization project. And I'll give you an example in a few slides. So first of all, let me switch to my first case study, which is the PI3 kinase delta inhibitors. So we arrived at quite some modification on the target at something which I describe now as compound A. Here you see, so each dot represents a single compound. On the x-axis is the lipophilicity as with logD, logP, and on the y-axis is the total topological polar surface area. And as you can see, this compound is in a great lipophilicity range. It's between 2 and 3, which we liked, but its PSA is quite high. And as you can see in this area of very high PSA, even though we're in the right lipophilicity space, we quite often observed that permeability is still insufficient to get decent pharmacokinetics in rat experiments. So what we wanted to do is actually come from this value and move it back down into this target area there. But we knew already at that stage that the target space between 2 and 3 in terms of logP was a good one. And here is why. Here you see bind logP values versus an assay shift we observed coming from the biochemical assay system into the cellular assay system. And here a brief reminder how to read box plots. So you see the median as the white line, and the upper quartile is depicted here. The lower quartile is here. So 50% of the values would fall into the box. The whiskers are 1.5 times the interquartile range, and the individual dots are outliers of this. And here you see quite clearly that we have a median of 10-fold assay shift going from biochemical in the cellular system or above. So 50% of the molecules showed a 10 or higher fold assay shift going from biochemical into the cell. So this was clearly not desirable. In contrast, we had another complicated or more complex assay version in a whole blood assay version where we observed the opposite. Notice here it's not as pronounced. The scales are a little different, but here you observe that the higher you go with lipophilicity, the higher you have the assay shift coming from the cell line assay to the whole blood assay shift. And here, of course, we could interpret it in the way if you're too hydrophilic, again, you will hit the permeability limit of the assay system. So this is of the cellular assay system. This was our interpretation. Whereas here, as you go towards more complicated assay systems, including higher plasma ratios, you've got higher unspecific binding to take into consideration. So this is the rationale here, and you see clearly that the logP range between 2 and 3 is the best area to be in terms of translating into cellular assay systems. And here is the whole discovery story coming from compound A to the final candidate molecule. I will talk you to this slide and the transformations we did. You can also look at progress with respect to number of aromatic rings or fraction of sp3 hybridized carbon atoms. The higher you are with number of aromatic rings, it has been shown that this is associated with low likelihood to make it. In the same way, a higher fraction of sp3 hybridized carbons is typically seen as advantageous in terms of moving forward. And this, if you look at this against time, you see that compound A is around here with 4 aromatic rings. And we made 3 crucial modifications, or actually 2 modifications that were quite instrumental for the success of the overall project. So first of all, we replaced this aromatic feature here, including one of the amides with this pyrrolidine ether linked, thereby, replacing an aromatic feature and one amide bond altogether. So this is compound B here, which we covered earlier, but had its impact on the project and later on. At the same time, we could also show that we could saturate one of the core cycles. This would be compound C. Again, you take away one aromatic ring, and you can see that you increase the fraction of sp3 as a consequence with respect to the compound where we started. And if you go along this time trajectory, you see we made marginal progress with respect to lipophilic efficiency. So the color code I should mention is lipophilic efficiency measured by the biochemical PIC50 and taking away our own Nibra logD. Again, color coded in a traffic light system. And if you combine these 2 features, all of a sudden you arrive at a compound class with only 2 aromatic rings and 50%. So this compound is exactly 50% contribution of sp3 hybridized carbon atoms, which was a landmark for the project because it didn't take too much time, and in fact it was between 5 and 10 molecules once we've identified this one that we arrived at the final molecule, which was our candidate and is now in phase 3 for activated P3 kinase delta syndrome. So again, here you see you increase your chances a lot with respect to being lipophilicity efficient if you take off aromatic features. If you increase sp3 rich features, you get into a much better property space, and this ultimately was the success for that project. So if you look at the target we set out to do to arrive in the target area, well, we did not quite achieve it, which I'm always quite proud of, because rules are never rules, they are guidelines, and we deal with probabilities and not absolutes in medicinal chemistry. So here you see that wonderfully we made the right call and roughly made it into the target space, but obviously we're successful with this one. This is the small vignette I wanted to show you on ROR gamma. So we started here with quite lipophilic compounds that had high potency. So here you can plot the logD versus the PIC50, and the individual lines here would respect individual lines with respect to lipophilic efficiency. We encountered a dead end if we wanted to just improve the logD. Here you see we're in a PIC50 range of roughly 6. So this is a micromolar compound that did not bear any cellular activity whatsoever. However, when we introduced this additional feature containing that carboxylic acid, we could show that we are doing this in a much more productive way, and could move into the LipE 5 space and ultimately arrive increasing the potency and still take off some lipophilicity with a compound like this very cyclic molecule where we could show that we achieved in vivo efficacy. So in terms of time, I will not talk about metabolism a lot, but I want to get a little bit towards forecasting those drug properties. And here's the weather forecast for today. I live in Switzerland, so my forecast is quite grim. But of course, this forecast is based on everything that we know before with respect to weather. And of course, there's nothing else in medicinal chemistry. And I will start with a question. So which parameters are important and unite successful oral drugs? And this question was first really asked by Chris Lipinski, at that time working at Pfizer, and answered in a seminal paper in 1997. And what is now the famous or infamous rule of 5. And it's not 5 rules, it's rules that where the individual value is can easily be divided by 5. So the C log P lipophilicity should be below B5, lower than 5 hydrogen bomb donors, lower than 110 acceptors, and molecular weight below 500. And the rule now says, and this is important, that poor absorption or permeability are likely if 2 or more of these parameters are out of range, and this is quite often forgotten. It's become hugely influential. As you can imagine, it's quite easy for parameters that medicinal chemists can easily relate to. We can design molecules that are based on these parameters, and the concept has had a huge impact. It's been sometimes religiously incorporated, sometimes with hard cutoffs, sometimes been used as a guideline, and it's been found that natural products were found to fall outside the rule of 5, and this was an important observation at that time. So common misunderstandings is in order to develop a successful drug, all of these criteria need to be met. So this is a too stringent interpretation, as you can see here. It's also been tempting to move close to these parameters in all of these areas, and this is also not a good idea, because these individual cutoffs are based on 90% percentiles, and of course you run into developability problems if you move towards this 90th percentile. So only 10% of the real drugs are above this percentile. And in recent years, it's been quite popular to revisit this rule of 5 in the context of today. And whereas the initial rule of 5 was based on compounds that made it to phase 3 and beyond, my colleague Mike Schultz in this Journal of Medicinal Chemistry article has looked at all the FDA drugs and that became new chemical entities. And here, this is the rule of 5 in the context of FDA approved drugs. So this is the 90th percentile. And how did it develop over the years? So you could say, okay, we became a little bit more lipophilic, at least with the 90th percentile, but what has significantly increased is the molecular weight. Whereas if you look at the median, the logP has certainly increased between 2 and 3, so this is quite significant, but overall has stayed below 5. And again, here you see the trend towards higher molecular weights. And out of all these rule of 5 cutoffs, if you think about it, molecular weight shows the largest time-dependent increase, and it's fueled quite a heated debate in the literature whether molecular weight is really something that you should look like and consider as a drug-like property. And I put drug-like here in apostrophes because what is drug-like is also sometimes a quite arbitrary definition. So what happens if you now move into this beyond rule of 5 space, sometimes abbreviated like that? You will encounter this quite frequently in the literature. But again, the beyond rule of 5 is as much misinterpreted as the rule of 5 itself. So how many rule of 5 parameters are outside this threshold? Remember, the rule of 5 said that 2 parameters should not be outside. But in many cases, I seem to think that the beyond rule of 5 seems to be associated with compounds that are of higher molecular weight. So as Alessio already alluded to, and Luc also, larger molecules become much more frequent in drug discovery. And this has 2 main reasons. So many of the current proteins that we tackle these days are large, flat, featureless binding sites where you need to cover a lot of ground in order to gain decent affinity, like many protein-protein interactions. But of course, also new modalities such as heterobifunctional degraders or PROTACs, as you heard. And of course, as you go higher in molecular weight, you also need to cover that with a higher hydrogen bond acceptor or polar surface area count, because you can't just add carbon atoms. You need to counterbalance these with a little bit of polarity in order to counterbalance that lipophilicity space. And this is just outlined. If you want to stay in the same logP range, you need to actually increase the hydrogen bond acceptors or the total polar surface. Again, this is the data coming from this PI3 kinase, that data set that I showed you beforehand. And one area that is problematic if you go to higher molecular weights is that you need to achieve a finer balance to get to both soluble and permeable drugs. So on the left-hand side, you see an analysis where solubility is actually largely unimpacted by higher molecular weight. Here you see the bind molecular weight buckets, and here you see with solubility range, color code, the bind property forecast index. So this is a GlaxoSmithKline forecast index, which is nothing else than the log-D plus the number of aromatic rings. And if you look at that area of 500, 550 plus, it largely seems unaffected. You should still stay below the PFI of 7 in order to get a good chance for soluble drugs. On contrary, if you look at permeability, typically higher molecular weights require higher lipophilicity in order to stay permeable. And this is quite often the problematic feature. And second way to look at this, which has become much more popular in the recent years, is the concept of dynamic lipophilicity. And you can see a cartoon on this on the left-hand side. So you need to look at multiple conformations or conformational ensembles, whereas one conformation might be good for solubility. You also need to have sufficient population of a conformation that is more lipophilic, that can make it through the membrane, and then once it's through the membrane, can exert again it's a conformation that is associated with solubility, and then also a conformation that of course leads to engaging the protein of choice sufficiently. In other words, this has also been looked at experimentally. If you look at, for example, compounds like this, PROTACs like this, you could look at NMR studies where you define 2 different, or you can find different conformations. If you look at polar solvents and apolar solvents, and the total polar surface area, this was one of the questions actually in the chat before, what is the total polar, the topological polar surface area required for PROTACs? This is an answer that you cannot give like that, because you need to take into account some of these intramolecular hydrogen bonds that lead to then a conformation that is good for permeability. And there are multiple ways to correct this polar surface area. One of them is the solvent accessible 3D polar surface area, or experimental polar surface area determined by supercritical fluid chromatography. And that's, I think, the way forward to really consider this dynamic lipophilicity for some of these PROTACs. And I would like to highlight one additional seminal work by AbbVie chemists, which is quite remarkable. If you look at the molecular weights of these compounds, these are compounds that have still oral bioavailability, and I think this is still, looking at these compounds is quite interesting to see that they made it. Came actually because there are 2 binding sites on BCL2 that they combined in order to get the necessary affinity, and this is their own forecast index, combining also then a number of rotatable bonds. So if you're too high in number of rotatable bonds, your fraction of compounds that make it to high bioavailability, which is binned here, goes down. So again, you should keep that as low as possible. And as you can see, this first compound that made it onto phase 3 still made it, is quite high in this context. But if you go lower, decrease the number of rotatable bonds, also decrease the lipophilicity, which goes into this multi-parameter score, you go in the right direction. And this is actually the compound that is improved for oral bioavailability. And just to finish off the science, so Arvinas, one of the frontrunners in the PROTAC field. This is just one example 140 out of their patent application. I chose this example because there's some degradation data on this one. So this could cover their frontrunner, which is ARV-471. The structure has actually not been disclosed yet, but has been shown to have in vivo efficacy in various estrogen receptor alpha dependent models at doses below 30 mg per kg. So this is quite remarkable, and could be one of the first compounds that make it to the market. And this is a compound, so this could be compounds represented by quite a high molecular weight. So overall, I want to finish with some personal recommendations. If you want to improve your Physchem, I would go for achieving the highest possible potency with the logP or D as low as your permeability limit allows. I would use lipophilic efficiency to monitor your optimization progress within a chemical series. And for ligands with higher molecular weight, I would argue, and I'm sure we get back to that in the panel discussion, I would say the game does not really change dramatically. Still, lipophilicity is the key parameter that needs to be controlled. Look at dynamic lipophilicity where required. Also, the permeability is the one area that probably requires some special attention. And for bifunctional degrader design, this is the teaser in a little bit, do you really need the high affinity on the POI? And actually, this was already almost answered by Alessio, that you perhaps don't need to have this high affinity. Remember, this is a catalytic process. You don't need to get too much into the cell. You need to just make sure the catalytic turnover takes place to a sufficient level. And if you don't need to be that potent, perhaps you can spare some molecular weight overall. So with this, I'd like to close. I'd like to thank both of the team members, where I was lucky enough to be a part of the PI3 kinase delta project team and the ROR gamma project team. Certainly a pleasure to be here with you today. Thank you very much for the invitation again, and I'm happy to take questions.
Thank you very much, Klemens, for your nice presentation. You nicely described the importance of lipophilicity. And I have a question related to the blood-brain barrier permeability of small molecules that sometimes is not so easy to predict. Can you comment on this? If there is a rule of thumb for small molecule to cross the blood-brain barrier?
So for small molecules, and I would not make the distinction again between small molecules and PROTACs too much. PROTACs are still low molecular weight, small molecules to me. But I would say you would like to be below polar surface area of 115 and probably significantly below. There's one of these property forecasts index that I mentioned in the list at the beginning. This is the Pfizer MPO multi-parameter optimization value. There a number of other parameters make it into their that have been shown to make it into successful brain uptake. So it's a little bit more challenging. Certainly I think for PROTACs, it will be, there was also the question in the chat earlier, it will be quite challenging to get there. But again, if you consider compounds like the SMARCA binder that Alessio showed at the beginning, perhaps in combination with a cereblon ligand and a little linker, you're not so far away from just a normal low molecular weight small molecule drug compound where you can tune these properties. And in addition, I would say that the more we learn about this dynamic lipophilicity concept, the better we will also be able to design these drugs to cross the blood-brain barrier.
Thank you very much, Klemens. We have much more questions, but we will discuss them during the roundtable in the interest of time.